U.S. commercial poultry meat industry is the biggest in the world. In the year 2010 alone, almost 37 billion pounds of meat were produced for a profit of $45 billion. In that same year, commercial poultry farmers lost 3.8% of their flocks, which could have been an additional $1.8 billion in profit. One organism that we know causes these types of diseases to poultry which result in this loss is Mycoplasma iowa. Now, we've known for a long time the different types of disease that are caused by Mycoplasma iowa, such as leg and joint problems, poor feathering, infection of the air sac, and death of embryos before they can hatch from the eggs. But we didn't know how the organism was causing this disease because we lacked a genome sequence for this organism. Our lab overcame that hurdle several years ago, and we found a few interesting things from the genome that may suggest how it might be causing this damage. The first was a full set of genes necessary to produce hydrogen peroxide, a mechanism seen by other similar bacteria to cause damage to their host. Of particularly interest to me was the finding of catalase, a protein that's known to break down hydrogen peroxide into water and gaseous oxygen. Now, this gene had never been seen before in mycoplasmas, and I wanted to see what impact this protein might have on the way these bacteria interact with their host. First, I wanted to look at the impact of catalase on the ability of these bacteria to produce hydrogen peroxide to damage their host. I measured catalase protein activity, hydrogen peroxide production, and the impact of peroxide production on the host using a laboratory animal-based model, and looked at catalase-positive Mycoplasma iowa against another catalase-negative species, as well as one engineered to produce catalase. And what I found is that when catalase is present, less hydrogen peroxide is produced, causing less damage to the host. This suggests that because catalase is present, Mycoplasma iowa doesn't seem to be using peroxide to damage its host. Second, I wanted to see if catalase was actually allowing this organism to be protective to its host in some way by neutralizing outside sources of peroxide, such as those normally encountered from the host immune system or from other bacterial pathogens that might be causing infection. I used the same laboratory-based animal model as before and incubated animals with peroxide and either catalase-positive Mycoplasma iowa or a catalase-negative species. Again, what I found is that when catalase-positive Mycoplasma iowa is used, less peroxide remains and more animals survive after 24 hours of incubation, suggesting that Mycoplasma iowa might actually be protecting its host in some way instead. And by continuing to study this protein more and some of the other findings from the genome of this organism, we can better understand how this organism interacts with its host and causes damage as well, leading to better treatment and prevention methods and less economic loss in the future. Thank you.